All right, let's go ahead and get started today. So hopefully you guys enjoyed yesterday's lab section. Most of the labs are going to be structured similarly, where you're going to be playing some kind of interactive game and hopefully, makes learning fun for you guys. A lot of those questions that I had on the worksheet are going to be similar to the exam, except it's going to be multiple choice. So I'm hoping you guys got a lot of those concepts across. Tomorrow's lab is going to be something similar, but it's going to be focused on the structure of DNA and then focus on stuff that I'm going to cover today's lecture. So today I'm going to be talking about the central dogma of molecular biology. So this is really important because this is how we become how we are. And so the general outline is I'm going to go over briefly what the central dogma is, like I did in the first lecture. And then I'm going to go ahead and review some of the structure of DNA and then compare this to RNA. And then we're going to go ahead and get into what a gene is, and then talk about the actual process of the central dogma, where we get into some of the details of what transcription entails, and then talk about how a protein is made from the transcription product. We're then going to go ahead and talk about the genetic code and basically how we're able to use this to figure out what kind of amino acids and proteins are made. Towards the end of the class, because I know some of these concepts might be a little bit difficult to grasp, we're going to have some practice questions, and I'll break you guys up into groups, and hopefully you guys can go ahead and assist each other when it comes to explaining stuff if anyone's struggling to understand some of the concepts. But the questions I'm posing are going to be basically some variation of this on the exam. So, take it seriously. And then I'm going to add I'm going to talk about point mutations and what this means for the genetic code. So what I want you to really take away from this is know what are the RNA nucleotides and what are their binding partners. Really understand what are the main differences between DNA and RNA, and be able to describe the steps of transcription and translation. And then from these products, from translation, I want you guys to be able to understand how to use the genetic code to decipher the amino acid sequence. And then the final thing I want you guys to understand is the 4 different kinds of point mutations that I'm going to go over and what they actually mean for how a protein functions. So we've been talking a lot about DNA this week, and we're just going to be talking about it in a different aspect. So yesterday we focused on DNA replication and the importance of why we replicate. It's basically when a cell needs to divide, it needs to transfer all of its genetic material to its progeny. So today, what we're actually going to learn about is what are these nucleotides in here? What does it even encode? Why do we have DNA? And so basically, it's through this process, the central dogma of central biology. So the first part of the lecture, I'm going to be focused on transcription, how a DNA molecule gets transcribed into RNA. And then the second part of the lecture is going to be focused on the translation process, how that RNA molecule ends up being converted into a protein. So let's go ahead and get right into it. So yesterday we talked about DNA, deoxyribonucleic acid, and it has a double helix structure. Well, when we compare this to RNA or ribonucleic acid, there are 3 main differences between the DNA and the RNA. So with RNA, it actually has, it's just 1 strand rather than it being double. The type of sugar that it has is different, which I'll go over into the next slide when we're talking about the structure of the nucleotide. And the RNA molecule uses all of the same nucleotides as DNA, except for thymine. RNA does not use thymine, it uses a uracil instead. So just be conscious of this. I'll go ahead and reiterate this point a couple of times throughout the lecture, but understand this because I will be giving you an RNA or DNA sequence. I'm going to ask you to make me a strands. Just be conscious of I'm asking you to make me an RNA strand, use the right nucleotide when trying to pick the right answer. So the structure of a nucleotide, I went over this yesterday. So both RNA and DNA utilize a 5-carbon sugar, which is known as pentose. So a DNA uses deoxyribose and RNA uses ribose. And so when we look at the structure and compare the two, they both have these pentamer structures, but what makes them different is this oxygen. So again, what I said yesterday with the name deoxy or without oxygen is why DNA has its name, deoxyribose, whereas ribose has the oxygen present, so it's just ribose. The nucleotide for RNA and DNA both have a phosphate group, so there's no differences there. And they both use nitrogenous bases. With DNA, which I went over yesterday, it uses A, T, G, and C. But on RNA, it uses A, U, G, and C. So it switches this T out for a U. So just be aware of this. You really want to know who are the binding partners. So yesterday I went over that A binds to T in DNA and G binds to C. But with RNA, it's a slightly different. With the G and C, it still remains the same. They both have GC content. However, that A ends up binding with the U. So again, know that these are who interact with one another. So for classification of these nitrogenous bases, there's really 2 groups that they're identified as: primidines and purines. So for the primidines, they're basically one-ring molecules, and that consists of cytosine, thymine, and uracil. And with the purines, it's consisted of 2-ring molecules, so adenine and guanosine. This kind of requires a little bit of memorization, but just to help you remember the difference between primidines and purines, if you remember one, then you'll be able to remember the other. How I remember purines is that there's 2 molecules to it. So I think because it has more rings, it's more pure. You can use that mnemonic if that helps you remember. But just know these for the exam. If I were to ask you, like, identify all the primidines, or identify all the purines, or which of the list on here is a purine, something to that example. So just be able to know what letters fall into which category. For study purposes, I have this summary slide for you. Basically encompasses material that we went over yesterday and today, kind of looking at the structure of the nucleotides and some of the key parts to where polynucleotides stick together, so 5' and 3' carbons, and then highlighting the types of sugars that make them different. Here, additional study material for you, highlighting the differences between DNA and RNA. So again, it really is this nucleotide difference, and it's this structure of where the DNA is double-stranded and the RNA is single-stranded. So what is a gene exactly? So a gene is the basic physical and functional unit of hereditary. This is the information that we pass off to our offspring, but we also inherit it from our parents. For every gene that we have in our cells, there's 2 copies, and we get each one from our parents. Genes, of course, are made out of DNA, and these are the instructions to make a protein. Humans have approximately 20,000 to 25,000 genes, which end up encoding for over 100,000 proteins. And we know this because of the Human Genome Project, which began in 1990 and ended in 2003. And this was a multi-billion dollar project. And we have the genetic information that we know and have been able to advance sciences exponentially because of this project. So I'm going to go ahead and talk a little bit about what the gene structure looks like, because it's going to be important when I'm referring to the actual process of DNA converting to RNA. And a gene structure can be very complicated. So I basically highlighted only the main things that I think are important for you to understand so you can understand the subsequent slides. So the first part that you really want to understand is this promoter region of a gene. So every gene is going to have this region. And this is where these proteins called transcription factors are going to bind to. And these proteins are really important for initiating this protein or this gene to get expressed so it can be made into a protein. Somewhere between the first exon and the promoter is going to be the transcription initiation sequence. And so this is going to be the sequence that tells the RNA polymerase, this is where you need to begin RNA synthesis, where they can start putting on RNA nucleotides to synthesize that messenger RNA. The gene is composed of two regions, exons and introns. So exons are going to be the information that your cell actually uses to make a protein. And between separating the exons are going to be these introns. And so scientists are still trying to understand what is the function of introns. I remember like 10 or 15 years ago, they would call it just junk DNA. But we're learning that there's some more biological function that they have that we're still not fully aware of what it does. But evolutionary biologists hypothesize that the purpose of having these introns in here is to help with promoting genetic diversity. Because if you were to ever take any kind of upper div genetics class, basically in a cell or in a gene, there's multiple exons, but not all exons are needed to make a protein. So having the ability to switch out different exons can promote genetic diversity in a population. And then the final part that I really want you to understand that this gene has is a termination region. So the RNA polymerase needs to understand when it's done synthesizing. And there's going to be a sequence at the end that is going to indicate that. So the DNA template, one of the strands of DNA is going to be used as a template to make the RNA. And again, I'm going to highlight again, RNA uses uracil. It does not use thymine. And so let's go ahead and, I'm going to layer it on just like I did with the DNA replication, because I feel like it's a very complicated process. And I feel like layering it on with more in-depth for every round of layering we do will help you guys better understand what's going on. So basically during the transcription process, the DNA molecule will end up separating, and this is going to be through those transcription factors that I was mentioning earlier. And so one of the strands of the DNA will be used to generate that RNA. I'm not going to really talk about what strand the transcription factor needs to initiate this in this lecture. When I talk about viruses tomorrow, I'll probably elaborate a little bit more on that. But just for simplicity and focusing on this, just understand that only one of the strands, and there is a designated strand that of the DNA template that is used to make this RNA. During the transcription process, there's basically three steps. There's the initiation, elongation, and termination. And this is going to be a common thing that we see in biology. So once the RNA is processed, it'll end up becoming a mature messenger RNA, which can exit the nucleus via the nuclear pore. Is there a question? Is RNA made the same way that DNA replicates? No. So I remember struggling to understand this concept, or I kind of fuse DNA replication with the Central Dogma, Essential Biology, separate those two. So everything that I talked about yesterday when it comes to DNA replication, that's just all the enzymes that are involved in just basically doubling the DNA was discussed yesterday. And the whole point of doubling the DNA, again, is only to give the information to the dividing cell. With the RNA, basically the cell ends up getting signals like make this gene or make this protein. In order to make this protein, the DNA and other enzymes end up getting signals to synthesize this RNA strand. So very two separate processes. And if you need more clarification, you can ask me after class. So let's go ahead and talk about some of these players. So the transcription factor, there's many different kinds. It's all really dependent on the kind of gene that's being made or expressed. And so what these transcription factors do, they have many functions. But for simplicity and trying to understand this process, what I want you to take away is the function of transcription factors is to facilitate the unwinding of the DNA and then recruiting that RNA polymerase. And so that RNA polymerase is going to be what adds on those RNA nucleotides to synthesize that mRNA strand. The transcription factors and the RNA polymerase together form this pre-initiation complex. So you need both of these in order for RNA synthesis to begin. If you don't have one or the other, then nothing's going to end up happening. So that's important to understand. So once these two fuse together, then, elongation can take place basically. And so what's going to end up happening is it's much more simple than DNA replication. All the RNA polymerase is using that DNA template strand to add on complementary RNA nucleotides to it. So you can see here, the RNA nucleotides present, highlighting again the U in the RNA, which it binds to the adenine. So just understand that that's the main function. And so when we kind of go through these steps, basically the RNA polymerase, along with the transcription factor, will bind to this promoter region, and that's initiation. That's how this synthesis of RNA strand is going to begin. And then the elongation step is just the RNA polymerase adding on those extra nucleotides. And then finally, the termination step is going to be when the mRNA is done being synthesized. And at the end of the DNA sequence, there is a sequence that is that encodes for the termination or the ending of the process. And so the RNA polymerase will be able to recognize this sequence and basically learn that or determine that this needs to, the replication, synthesis is done, and then it detaches. RNA is very unstable. It's a very unstable molecule. It gets degraded quite easily from nucleases. So nucleases are just enzymes that basically break up RNA or DNA. So if it's a DNA nuclease, it's going to fragment and destroy DNA. If it's an RNA nuclease, it's going to do that to RNA. So before the RNA even leaves the nucleus, there has to be a couple of modifications that are made to this new synthesized RNA strand, so it can be stable and not get degraded from other enzymes that are within the cytoplasm. So once the mRNA is, once the RNA is synthesized, what's going to end up happening is there's going to be some enzymes that are going to add this methylguanosine cap or a G cap at the 5' end of the RNA sequence. And then another enzyme is going to add a polyadenylated tail. So basically it's just a long sequence of A's at the end. And so the RNA molecule having this G cap and this poly A tail at the end is going to tell enzymes in the cytoplasm, don't degrade me, I have some kind of function. And so the final step that the RNA needs to undergo is splicing. And so as I mentioned before, exons are what encode for the actual protein product. The introns don't do any of that. Again, scientists believe that they play a role in promoting genetic diversity, but we still don't know what they do. And so in that process of splicing, basically the introns are removed and then the exons are spliced or stuck together to form one cohesive unit. Once all of this occurs, so this 5' cap, this polyadenylated tail, the introns are removed and the exons are stuck together, you now have a mature messenger RNA. And this can now leave the nucleus safely. Again, these exons are going to be what encodes for the protein. So we just went over what the main steps of transcription are. So now let's go ahead and talk a little bit about translation. So in translation hold on, there's a question before I move on. You said modification prevent the RNA from being destroyed by, I didn't specify what they get destroyed by. Lysosomes could be an organelle that that causes degradation. I'm not 100% sure, and I can actually look that up at what enzymes in cytoplasm destroy RNA. Yeah, I'm not too sure. So I can look that up and I can let you know for next class. But I'm sure lysosomes will play a role as they like to destroy things. And as you guys learned from yesterday's lab. Okay, so in translation, this messenger RNA is going to serve as the instructions to make a protein. And the way this is going to be done is by the system reading these things called codons. And so codons are just these 3 letters, these 3 nitrogenous bases that are all going to encode for one kind of amino acid. And so these amino acids are basically going to be the building blocks of the protein. And so this is all going to be done at the site of the ribosome, depending on if it's the ribosomes in the cytoplasm or the rough endoplasmic reticulum will determine where that protein ends up going within the cell. But I know people kind of get confused with like amino acids and peptide, polypeptide, and protein. They're all more or less talking about the same thing. It just is dependent on the level. So think of the amino acid as the fundamental building blocks. And then a peptide or polypeptide is many peptides stuck together or many amino acids linked together are going to be what make up this polypeptide. And basically, I think after a series of 50 amino acids are strung together, that's technically considered a protein. So it's all really nomenclature, but they can be used interchangeably, but it's all dependent on what unit they're talking about. So amino acids, main blocks, them stuck together are a polypeptide, and multiple polypeptide chains together make a protein. Okay, nice. Somebody. Thank you, Brianna. So in order for translation to take place, there are 3 different kinds of RNA that are used. And the first one, of course, is our messenger RNA. That's what we need. That's going to have all of the information that's going to encode the protein. And that's all going to take place in the cytoplasm or the rough ER within the ribosomes. The other kind of RNA you need are these transfer RNAs. So here we have it highlighted in green. And so these molecules are going to carry the amino acid of interest. And there are many different kinds of transfer RNAs, and it's all dependent on what the anticodon it has attached to it. So the anticodon is basically going to be the complementary sequence to whatever is the codon on the mRNA. So they have to match, and you'll get more of a sense of that in tomorrow's lab games. And then the final kind of RNA that you need to make synthesize your protein is ribosomal RNA. And basically, these ribosomal RNAs are going to facilitate the binding of the mRNA to the actual ribosomal unit. and also play a role in creating polypeptides when the messenger RNA is actually being translated. So let's go and talk about some of the key structures within this translational mechanism. So let's start off with the ribosomal structure. So the ribosome is composed of a large and small subunit. The mRNA binds to the small subunit, while the transfer RNA will end up binding to the large ribosomal subunit. Within the ribosome are 3 different binding pockets. And so basically, these are going to be the sites where all the translation process is taking place. So in site A is basically where your mRNA is going to come in and get read. And then, so it's going to be the entry site. And then after it moves to the next location, the P site, this is where the actual polypeptide chain starts to get synthesized and catalyzed. And then once the amino acid from the transfer RNA is placed on the respective polypeptide sequence, then the transfer RNA is going to exit here. That's what the E site is, as well as your RNA is going to continue to come out through this end. With the transfer RNA structure, all transfer RNAs have one specific amino acid that's associated with it. And so here I have a 3D rendering of what a transfer RNA looks like. And then here I have a 2D simplified version. And so here we can see where the amino acid attachment sites are located. And then on the opposite end is going to be where your anticodon is located. And so these 2 parts of the transfer RNA are really important to understand because they both facilitate the translation process. And so again, this anticodon is going to be the complementary sequence to whatever is on the messenger RNA. When protein formation begins, the first anticodon that needs to match for the protein to actually begin is encoded by a start codon. And the start codon is going to have the sequence AUGs, so adenine, uracil, and guanosine. And this sequence is going to encode for the amino acid methionine. And every protein is going to begin with this amino acid. Once the elongation process is over and the enzymes or the transfer RNA wants to know that it's done synthesizing, or sorry, it's done translating the mRNA and making the polypeptide, it will end up encountering a stop codon. And so with stop codons, there's 3 of them in total. And so these are what they encode for. I don't expect you guys to memorize these. If I have you guys encode or try to figure out what these sequences mean, I'm going to give you a table so you go ahead and decipher them. And I'll get into that in a couple of slides, and we'll go ahead and get some practice questions with those. All right, I think that's all I wanted to say there. So the actual translation process. So just like with the transcription process, the translation has three parts: initiation, elongation, and termination. And so, like I mentioned in the previous slide, when the initiation begins when the transfer RNA brings the first amino acid to the P site of this polypeptide chain. And that, again, is going to be methionine. Once this basically begins, the whole elongation process begins, where the mRNA is going to continue to get fed through the ribosome, and transfer RNAs are going to come in and basically identify the complementary sequence. And then basically ribosomal RNAs are going to facilitate the catalytic addition of the amino acid that is coming in from the new tRNA and adding it to this sequence. Once the mRNA is all read or translated, it'll run into a stop codon. And again, there's 3 stop codons that are basically going to tell the translation machinery, you're done synthesizing the protein, go ahead and ship it off to the Golgi apparatus so it can get folded properly and exported to wherever it needs to go. So here is a list of amino acids. I'm not expecting you to memorize them, but just to kind of have a look at the important ones that biological systems or organisms need to live. And so here's basically the key chart for the different, I guess, phenotypes or structures of these respective amino acids. So go ahead and have a look at them on your free time. Yeah, somebody asked me, where does the transfer RNA come from? So they're floating around in the cytoplasm. I don't know how close, I'm assuming that they're relatively close to where the endoplasmic reticulum and the nucleus is at. Where we actually like got them from, I don't really know the answer to that. I know there's this process of what is it? Is it symbiosis, where we basically, how cells kind of came to be. The reason why we have a lot of membrane-bound organelles is because they cells basically started eating each other or phagocytosing, and then some of those things were able to survive, and then it ended up getting complex organisms like ourselves. And if I didn't answer your question, feel free to find me after class. So here is the genetic code for translation. And so you guys do need to understand how to use these. So I will probably give you both of these, or I'll tell you guys on the test, like make sure you guys have these open so you can go and use them when it comes to translating a sequence. And so the way you use this is, I'm going to end up giving you a sequence, and say the sequence is CUG. And I want you to tell me, what amino acid does this encode? Basically, to use this genetic deciphering code on the left, you basically will start from here. So we'll start with C, and then we'll go to U, and then we'll go to G. And so we can see that the amino acid that this sequence encodes is leucine. And we can go ahead and apply that same concept to this other graph. So we'll start with, on this end with C, and then we will go to this column here for U. And then when we look for the G, we see that it encodes for leucine. It's important to understand too that there's multiple sequences that encode for the same amino acid. And this is going to be important when it comes to genetic mutations that occur that I will end up getting into towards the end of the class. And so here we have the start codon that I was telling you about earlier, AUG, and it encodes this methionine. And this will always be how proteins or polypeptide chains begin. And then here we have the respective stop codon. So these stop codons do not encode for an amino acid. It just tells the machinery to stop synthesizing the RNA. It's done. Go ahead on to the next process. And so basically what I'm going to do is break you guys out into rooms again. I'll give you guys about 10 minutes. But what I want you guys to do is basically practice translating these RNA sequences and picking the respective amino acid that corresponds to whatever the sequence I have present. And then, so I'm gonna have you do these 4. And then I also want you to do these 3. And you guys should have all access to the questions online. So I don't think you guys are gonna be able to see my slides. once I break you guys out. So maybe somebody can pull it up during the your breakout room, or just pull it up on your screen. But this one's going to be a little bit different. It's asking the same thing, but in a different way. And so this is going to kind of cause you to think a little bit. And so the question that I'm going to have is, if this tRNA carries some kind of anticodon, I'll give you that sequence. I want you to tell me what is on this end. And you're basically going to have to work a little bit backwards. And just remember that the genetic code uses codons to decipher amino acids, not anticodons. And so I'm giving you an anticodon, so you're going to have to work backwards, figure out what the codon is, and then use that codon to figure out what the amino acid is. Do you guys have any questions about this before I break you guys out? All right, I'm going to stop sharing this for a second, and let's go ahead and break you guys out. 1 second. 66. All right, I'll see you guys soon. I'll give you guys about 10 minutes, and I'll make an announcement once we're all good to go. And Brianna, thank you for helping me answer some of those questions. It'll save me time later.
No problem, no problem.
Are the people that are left in the breakout room, are you guys having trouble moving over to your group, or are you guys not present at your computer? Feel free to unmute yourself.
Oh my God.
Oh, hello.
Yes, you asked for some help.
Oh yeah, I had a question for number 2.
It's supposed to be the MET amino acid, THR, and then it's supposed to be lysine, but I don't see the answer on there.
Oh, did I mess up on something? 1, 2, 3, 4. Oops, I might have messed up on that. 1, 2, 3, 4. Well, sorry, which amino acid was it?
Shouldn't it be MET, THR, and then lysine as the third amino acid for number 2?
Oh, the third. Let me look at the code really quick. Triple A, I think so. A. Yes, did I fuck up on the answer? My mistake.
Yeah.
Thank you. I will go ahead and make an announcement. If you guys catch any other errors, please let me know.
Okay, thank you.
Thank you. I might have yeah, let me see, because I might have forgotten to delete it. What's GAG? Here, I'll let me do this one really quick.
Okay.
Let me see.
Acid does a tRNA.
That's right. G A. Let me go ahead and make an announcement to the class just so people are aware and I'm not getting the same question.
Okay.
But yeah. We'll go ahead and disregard that one. Yeah, if there's other errors, please let me know. Thank you.
Thank you.
Okay.
Did I mess up again?
Yeah, you did. Number 6, it should be TRP.
Okay, let's see. Number 6. All right.
I think.
Let's see. I might have been, okay, let's see. ACC, so we're going to convert that. So ACC goes to UGG. U, G, 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 yes, TRP. What did I put? I put TYP, TRP. Yes. Thank you. All right.
But everything else, yeah, it's fine.
Okay, perfect. Well, I'll make another note. Thank you so much.
You're welcome.
All right. Hopefully you guys enjoyed that. Sorry about the mistakes. There was two, sorry, lettering. So let's go ahead and see how everybody did working together. I hope it was pretty straightforward. You will get a question like this on the exam or a couple of questions. So if you guys are struggling to understand this concept, please come see me after class. So go ahead and put in what you guys wrote. And let's see how people are understanding this. Are you guys able to see the poll? Oh yeah, good. Did people need more time to work on these questions? Go ahead and let me know in the chat. And I think once we, right, all right, well, okay, that's fine. So as long as you're understanding the concepts behind it, let's see how most of the class is doing with these answers. And if you guys are doing well, I'll go ahead and just move on. If people are still struggling with some of these, we can go ahead and stay after class and kind of walk through it a little bit. So we're at 36 of 65 students. So I will probably try to get hopefully about 50 of you guys. Yes, so I don't know if you guys got my broadcast message. I messed up on question 2. I don't think I had the right, I translated one of the proteins wrong. So disregard 2, don't even worry about it. I will go ahead and update it. I've already updated on the slide, and I'll go ahead and update those on iLearn just so you guys have them. But I believe the answer that I originally had for that one was D. Yeah, it was exactly. I misplaced the those. I switched those two, so it's supposed to be lysine. But good for rationalizing. If I end up messing up on a test like this, then that question will be thrown out. But let's hope that I will double check ahead of time. Yes, and then I also broadcasted that one. A should have been tryptophan instead of tyrosine. So yes. And I've edited those already. So yeah, sorry about that. All right, so we are at, I'm going to end this poll, share the results with you. All right, so yes, number 1 was B. 92% of you guys got that, so that's good. Number 2, I fucked that one up. Sorry about that. So yes, disregarded, but it should have been D. I think some of you guys rationalized that with the one error. Number 3 should have been B. 70% got that one right. Can anybody tell me what you guys were struggling with that one, or was it just translating it wrong? Anybody? All right, we'll move on. Number 4 should have been C's, okay, 74%. Number 5 should have been B, 68. Okay, so we're roughing around 70% on that. All right. 6 should have been A, and again, that was my fault, my mistake. It should have been tryptophan. And 7 was D. Okay, 66%. Okay, so I'll go ahead and pick one question from each, just to kind of hopefully, hope you guys understand this concept. And then if you guys are struggling to understand still, come to my office hours or come after class, because there should be some time. So let's see. So the tRNA amino acid at the top is whatever bonds with the, so the tRNA amino acid at the top is whatever bonds with the anticodon. No, so the tRNA has 2 parts to it. So there's the amino acid at the top, And then there's the anticodon at the bottom. The anticodon is what's going to recognize the codon that's on the mRNA. And so we can go ahead and start with that, just to go ahead and, let me close this real quick. 1 second. So we'll go ahead and start with, we'll do the one that I ended up messing up on, just so we all are on the same page with that. Let me share this. Let me actually record this. Oh no, it's a recording. Good. All right. So, for 6, so I tell you that the amino acid that's attached to this transfer RNA, or you guys are trying to figure out what that is. And so at the bottom of this is ACC. That's the anticodon that this tRNA carries. So let's go ahead and write that down, ACC. So what you have to do is work backwards and figure out what is the codon to it. And so you have to understand who pairs with what. And so we know that A pairs with U when it comes to RNA. If it's DNA, it's T, but we're talking about RNA here, so it's U. And then C will always pair with G. So we have UGG, okay? And so now we're going to want to use that code to figure out, okay, what is the amino acid that goes along with that. So if we start with U and then go to G and then go to the other G, we see that it's tryptophan is the answer. Does that make sense? Okay. So hopefully that concept is pretty straightforward. It just requires you to kind of think a little bit about the structure about it, but they're going to be pretty straightforward. All right, and then let's see. Let's go through another one. So let's go ahead and pick the other one that I ended up messing up on. So let's do 2. So 2 is supposed to be D. I've changed the lysine to its proper, or the right amino acid here. So basically with these sequences, all you're doing is using that genetic code to decipher what these are. So the questions 5 through 7 had you work backwards. This one is just very straightforward. So if I have AUG present, I'm just going to go ahead and use the code. So I already know that AUG is going to code for methionine. And then let's see, ACU, ACU, so that's 3 and 9. Okay, so that's there. And then triple A, lysine, GAG, so glutamic acid, UCA, serine, and then this last one is going to encode for the stop codon. So I'm hoping that that makes sense. If you guys need some more clarification, office hours or after class, but they're for the most part pretty straightforward. I will always give you guys this code. I'm never going to ask you guys to memorize any of this. That's ridiculous. Oh, somebody asked me if I could do 7. Sure. Let me see what 7 looks like. All right. So UCA, unless I messed this one up, but I think to my knowledge, I only messed 2 of them up. So the same concept. I'm giving you guys a transfer RNA, and I tell you that the anticodon is UCA. So UCA. And I'm trying to figure out what amino acid is attached to this. So the anticodon is going to be present here. So I need to basically find the complementary strand to this. So A is going to bind to U. G is going to bind to C. And A is going to bind to U. So my codon that my anticodon from my transfer RNA is going to bind to is coded for AGU. And so if I look at AGU, I see that it's serine. And is serine actually the right? Yeah, so answer is D. Does that make sense? Okay, good, good. All right, so we'll go ahead and move on since we're running out of time and I have a couple more concepts to go over. Again, find me after class if none of this makes sense. So I'm gonna go ahead and take a poll. So we're gonna talk about some genetic mutations, but I kind of wanna gauge you guys' I guess, knowledge on understanding the differences between us and other species. So I'm gonna go ahead and do another poll. And stop sharing. Okay, where are you at? Sorry, there's only one to this. I don't know why this other one messed up. So how different is your DNA from our closest ancestor, the bonobo chimp? I'm just interested to see what you guys think. And then I think it makes for fun discussion. And I'll go ahead and wait until the majority of the class. I'll wait for at least 50 people to see how close we are. So how much of our DNA is similar to that of the bonobo chimp? Oh, I go ahead and put a random answer for number 2. Sorry about that. Can somebody explain to me what are you saying? Monkey, or is there does that mean something? Oh, a meme. Got it. Feel free to share it with me. I'll try to include some memes in future classes. All right, so 57 of you guys have given your answers. So let's go ahead and share these results. So interesting. So I feel like a lot of you guys don't really know. So the answer C. It is 1 to 5%. So it really depends on the species of chimps. So the bonobo chimp, it's estimated that we're 1% different from them. So we're for the most part the same. If you compare our genetic material to the common chimp, it's going to be closer to 4 or 5%. And then I'm going to have one more fun one because I'm curious. So now that you guys know that, let's see what you guys say for this. So how different is your DNA from mine? Let me launch another poll. Somebody asked how much DNA we share with bananas. If I'm not mistaken, it's around 50%. Yeah, it's crazy. We're all connected. Evolution is a thing. All right. Yeah, we share a lot of things with a lot of organisms that exist. All right. 57 of you guys have answered. I'll give you a couple more seconds. 3, 2, 1. All right. I'm going to end this poll and share results. So the actual answer is D. I don't know why it's highlighted red on E here. But the answer is D. It's 0.1%. So humans, all of us, are basically the same. It's these small mutations that we have in our DNA that make us distinct from one another. Let me go ahead and stop sharing this and closing this poll. All right. And so I feel like with everything that's been going on in this past year, and me being a biomedical scientist student, and I think it's important to address this issue of racism. And it's important to understand that racism is a social construct. It's meant to divide us. It's not, there is no biological difference between any of the humans that are on here. We aren't distinguished by different species or subhuman. We're all the same. It's just this very small percentage of DNA that's different that makes us different. We are all one human race. And to kind of, emphasize this thought, we all have this gene called TYR. And this gene is encodes for an enzyme that's called tyrosinase. There we go. This gene has it has a couple of different functions. It really plays a role in what our hair, skin, and eye color look like. And tyrosinase is the first step in producing the skin pigment melanin. And melanin is going to be what our skin color looks like. And evolutionary speaking, the whole point of having this protein in our skin is to block the sun, because the sun releases radiation, UV light, and it's going to create DNA damage to our DNA. And we don't want that, because that's going to lead to mutations and potentially cancer. And so humans that live or come from sun-saturated regions, such as different parts of Latin America, different parts of Africa, different parts of Southeast Asia, they tend to have more melanin because they have more of this enzyme being produced. And so with that, you're going to end up having darker skin. Those people that live in a sun-limiting region have lighter skin. And it has to do all with the whole pathway and how this gene gets upregulated and how this enzyme produces to make more or less melanin. And it's all because of the sun. There's a link in the notes section that I highly recommend that you guys watch. It's very informative of talking about, it talks a little bit more about some of the science as to why we have different colors. They cover some of this, but then they also talk a little bit about the effects of the sun and melanin and vitamin D. But I just wanted to throw this out there because I feel like it's a really important topic that needs to be covered, because there's a lot of different, there's a lot of people that still believe that we're all different races, and it's something that the government created to distinguish people, but we are all the same. So please know that. So how do these DNA mutations occur? So there are spontaneous mutations. So these, happen naturally. As I mentioned yesterday, when the DNA replicates, we have the DNA polymerase that's adding on all those extra nucleotides. And every 10 billion nucleotides that it adds, it makes one mistake. And sometimes it's not able to fix that mistake. And so then you end up getting a random mutation in that fragment of DNA. The other kinds are induced by some kind of mutagen. And so the two main types that I want to cover are high energy radiation. So this is really going to be from X-rays or the sun. And so that's why it's really important to wear sunscreen, because these the sunlight ends up causing this dimerization of nucleotides together, and that is going to create problems when you're trying to express proteins and whatnot. So yeah, wear your sunblock. And then the other one, which I find interesting, are the chemicals that are around us. We live in a very dirty world, unfortunately, and chemicals that are going to produce mutations in our DNA are going to be examples of smoke, be it from cigarettes or from smoke coming out of factories. That's going to contribute to dirtying our air. And the one that I really wanted to focus on is BPA, because I feel like a lot of people don't really, like, they hear about it, they know that it's found in water bottles, but it's also found in your receipts. And it's also found in canned foods. And this molecule is not only going to lead to DNA damage, but it also we're now learning that it's leading to infertility because it acts as an endocrine disruptor. And so with the endocrine system, that's all hormone related, a lot of your, when it comes to regulating the sperm and the egg, a lot of the, there's a lot of hormones that are implicated in that. And so if you end up messing that up with some BPA in there, potentially you're not going to be able to have kids, or there's going to be issues with them. So please wash your hands after you touch any receipts. I think it's good to know. So yeah. So let's go ahead and talk about how a mutation in DNA can lead to some effects. So a mutation in any change in the DNA sequence is going to be considered, it's going to be classified as a mutation. And there's a few different kinds. So there's replacing one of the letters, there's deleting or adding a letter. And this can have a wide range of effects, really dependent on where this mutation occurs and what amino acids it's going to encode for. Mutations don't have to be bad. We've been mutating and adapting basically since however long ago, since our species of 200,000 years ago or something like that. And even before, you know, we've been evolving from other creatures. So this is a natural thing, but it's really dependent on the source of the mutagen that leads to this, this change in the nucleotide. And that's going to determine how harmful a lot of this is. So, so what might happen to a protein product if there's a change in the nucleotide sequence? So we'll go ahead and use the example of a sentence just to get this concept across first. So in a sequence that has no genetic mutations, we have a normal sentence: the cat ate the rat. Now, what happens if you have a point mutation that changes one of the letters? So now we have an R, we have an H that was originally an R. So the cat ate the hat. Grammatically, it still makes sense, it still functions, but it kind of doesn't make sense a little bit. And then what happens if you were to delete the C? Basically, everything's going to end up moving over, and there's just going to be a lot of, there's only a couple of words here that actually make sense, the and her. And then what happens if you add a letter? You're going to end up pushing everything over. And again, there's going to be more shifts that occur within your sentence. And so the meaning of all of these sentences basically is gibberish. There's going to be problems with you trying to express what you're saying. And this is no different when it comes to the point mutations that occur in our cells. So here we'll take the example of ACG. And so in this fragment that has no kind of mutation, it'll get converted to RNA and it'll encode for cysteine. Now we have this other kind of mutation called a silent mutation. And basically what ends up happening with these kinds of point mutations are one of the letters are changed, but it still encodes the same protein. So there isn't any issue. So going back to that genetic code slide that I had showed you guys earlier, there's basically multiple codons that encode for the same thing. So sometimes there isn't going to be an issue. Then you have these other mutations called missense mutations. And basically this is what, when you end up changing one of the letters and it encodes for a completely different amino acid. And going back to the amino acid chart, depending on how similar the amino acid looks will determine how well the protein functions. So say my protein or my amino acid originally without any mutations encoded for aspartic acid. And I had a mutation and the protein ended up becoming glutamic acid. Well, the structure of these and the charges and everything are relatively similar. So more or less, the protein's going to function okay. Now, say my healthy DNA encodes for aspartic acid, but encodes for something else, say lysine or serine. The structures are very different. The charges are very different. Having these kinds of changes could potentially lead to the inhibiting the function of the protein. And that's how a lot of diseases end up occurring. So the last or third mutation is a nonsense. And basically with these kinds of mutations, a premature stop codon is made. So you have your sequence of mRNA, and if you end up making a stop codon right in the middle of it, it's basically going to stop synthesizing that protein. And if you can't synthesize it, then basically the protein's not going to work, and then that's going to lead to disease. And then the last mutation I want to talk about is a point mutation, frame shift. And this is another kind of point mutation. So this can be either an addition or subtraction of a nucleotide. But what's going to end up happening is if it's an addition of a nucleotide, it's going to shift it one way. Or if you delete one of the letters, then it's going to shift it the other way. But what ends up resulting from a lot of these frame shifts is amino acids that are not anything like what they originally were supposed to be. And this is going to end up leading to defective proteins. So what I really want you to know are these different kinds of point mutations and what implications that they can have. If you guys have any questions, let me know. I know I'm over, right on time, perfect. Let me go ahead and give you guys the sign-in sheet, and you guys are free to go. And if anybody has questions, feel free to ask. I'll stay on here for a couple more minutes. Chat. All right, let's see. I got a series of questions in here. Is a mutation good? I think it's really dependent. It's really dependent, honestly. I would say if you're getting a mutation due to high levels of radiation, I don't think so. But if it's a mutation for for some kind of fitness, for example, like people that migrated from Africa to Europe, there was less sun in the northern hemisphere. So random spontaneous mutations basically made it so the skin would lighten up, so more sun could penetrate the skin, so that they can go in and get more vitamin D. So it's really dependent. Yeah, isn't there an HIV resistant one too? Yeah, I'm actually going to talk about that in, I think, 2 lectures from now. Maybe I'll include it tomorrow. I don't know. But yeah, so briefly, and I'll end up covering this again because I think knowing about HIV is pretty important. All viruses, and I'm going to emphasize this tomorrow, need a host cell to replicate. But in order for it to replicate, it needs to get into the cell. And so when it does this by binding specific receptors that are on the cell surface. And so with HIV, there is a population, I think it's somewhere in Scandinavia, somewhere in Northern Europe, where a very small population has a mutation in these receptors where they don't get made, or they're folded improper. Yeah, they're folded wrong. So if the proteins or these receptors are folded wrong, that means HIV can't bind to it. And if it can't bind to it, then it can't get into the cell. And I'm going to talk a little bit about how people are using this technology called CRISPR-Cas9 to make these designer babies to basically knock out some of those receptors. So they're basically HIV resistant. I personally don't think that's a good idea, just because those receptors that they're trying to knock out are really important for immune cell regulation and migration. But we'll talk about that. It's going to be an interesting lecture when it talks about some of the ethics to that. Okay, I got another question. For lecture quiz number 4, is it a multiple choice or are there 2 answers? Yeah, so I messed up on the, for yesterday's lecture quiz. It's one answer for everything. And I know a couple of people have missed this question. So if you have any questions about it, I can go and walk you through right now if anybody lets me know. Yeah, it's only one answer. And you really want to rationalize and get this concept of what happens to telomeres every round of replication that occurs. Happen on several cells at a time or at one cell. So I got a question. Do mutations happen on several cells at a time or on one cell and then spreads to others? So a lot of the time with mutations in our bodies, it depends on the kind of mutation, right? So if it's a mutation where the protein functions fine and everything, nothing's really going to happen. Now, if it's a mutation where it's going to lead to like DNA mismatches, or it's going to have problems with encoding the right proteins. Going back to that cell cycle pathway I was referring to, the cell host machinery is good at catching those. And so to prevent a cancer cell from forming, if it catches one of those cells, it's going to end up killing it, or apoptosis or programmed cell death will end up getting initiated. Now, if it's a mutation in one of those checkpoints where you have one of those proteins that regulates it, then basically that information is going to get passed down to the next daughter cell, and that's how you start to develop cancer. So in theory, mutations could be, yes, in theory, mutations can be beneficial, but I would say the majority are not, they're harmful. Can you go over Is bad again? Yeah, so why is BPA bad? So I don't know the whole actual mechanism on what it does. I know it does, I'm not sure how it actually, and I can actually look this up, DNA damage. I know that it does something to the DNA which promotes cancer, but what I was going over is BPA acts as an endocrine disruptor. So its structure is similar to some hormones that we have in our body that basically regulate themselves. And so if you end up messing any of that pathway up, then all your other, because there's a lot of, they're called feedback loops. So if one thing gets made, then it's going to regulate something else, and that's going to regulate this. And once it reaches a threshold, then some things will stop. And so with BPA, if it messes up something in that pathway, it's going to throw everything out of whack. And so it's known that it's playing a role in basically the hormones that are involved with sexual reproduction. And so that's where I go back to saying it's an endocrine disruptor. All right. I can go. So I'm on my phone. It won't let me sign in. I'll give you a 10 attendance points, so don't worry about that. Lecture quizzes are due Friday. Yes, all four. So DNA polymerase builds from 5' to 3' and moves. Okay. Yes, for the most part. So DNA polymerase moves from 5' to 3'. So it's building in the 5' to 3' direction, but it's moving in the 3' to 5' direction on the template strand. Does that make sense? I hope that makes sense. All right. I think that's all the questions. So if you guys have any questions for me, feel free to ask.
Wait, sorry, I had a question. So it moves from the 3' to 5'.
No, it moves from the 5. So here, let me pull up the lecture so it'll be easier for reference. 1 second. slide would be best? Okay, so I'll start with this slide first, and then I'll talk about the polymerase. I'll show this. All right. So just to get this concept across, basically there's DNA is read from 5' to 3'. And so its complementary strand is going to be the opposite side. So 5' to 3'. So DNA is, they're anti-parallel, so they're opposite of each other. Now, when we talk about DNA polymerase, let's find a good slide, this one. So if we look at how DNA polymerase functions, notice how this newly synthesized strand, the daughter strand, is from the 5' to 3' direction. So this enzyme right here is adding on nucleotides in the 5' to 3' direction, okay? So that's how it moves. That's what it's moving on. Now, when you're talking about what direction is it moving on the, parent strand, it's moving, starting from the 3' and then going all the way to the 5'. Does that make sense? Because DNA is when they're binding to each other, they need to be anti-parallel. And so the DNA polymerase is always going to move from the 5' to 3', which means that on the template strand, it's technically moving from the 3' to 5'. So it's all dependent on like what strand you're referring to. And so whenever I'm referring to the DNA polymerase moving in a direction, I'm talking about it making the new daughter strand. So it's always going to make the new daughter strand from the 5' to 3'. Does that make sense?
Oh, okay, I got it now. Thank you.
No problem. So we'll always be given a base strand. Yes, always. You will always be given a base strand, and I will tell you what direction it's in. And so just make sure that I'm gonna usually note what direction it's in. So it's always gonna be opposite. So if I give you a sequence that's from 5' to 3', when I'm looking for the answer in the multiple choice I give you, basically I'm looking for that complementary sequence that's going to match those base pairs, but I'm also looking to have the denotation of the 3' and the 5' where that belongs, and it should always be opposite of whatever the base strand or template strand is. Any other questions? No problem. Xing Yu, are you there?